Now let us look at some of the phenotypes of normal and cancer cells. Before going into that, we should know some terms. What is called anaplasia? It means lack of differentiation, because the cancer cells are poorly differentiated. I am sure you know what is differentiation, right? All of us are born with the fusion of sperm and ovum that leads to the zygote. When you look at the zygote, it looks like a mass, no visible structures and functions or anything like that. It looks like that, a simple cellular mass or tissue mass. But then when the fetus develops, you start seeing nose, eyes, something like that. Some structures are visible, some functions are, you can deduce them. So, this kind of a transformation of acquiring structural and functional characteristics is called differentiation, right? But cancer cells are poorly differentiated. So, that is what is the lack of differentiation is called anaplasia. Then a loss in uniformity of the individual cells as well as a loss in architectural orientation. Later on I will show you the abnormal structural changes that occur in cancer cells in the beginning, that is called dysplasia. There is another term that two terms that you must know. One is called hyperplasia, which means increase in cell number. It is not the same as increase in cell size. So, there are two terms that are used. Hypertrophy refers to increase in cell size, hyperplasia refers to increase in cell number, of course regulated and very physiological phenomenon also. Metastasis, I already told you that it is the cause for 90% of the deaths in cancer patients. It is the condition in which cells from the original primary tumor, wherever it is originated, migrates to either local, that is nearby, or distant locations in the body where they will divide and form secondary tumors, and this is known as metastasis. This is the most dreadful condition, and it is difficult to treat such patients, and people are also working on why this is happening, what are the mechanisms. So, a lot of research is going on to understand how the cells are migrating, why they are migrating to specific sites. We are coming to that probably in a few more slides, we will be talking about that. Now, normally the cell let us say divides. Let us imagine we have put some cells into a petri dish, something like this. So, it starts dividing and forms a layer. So, one by one they get attached and there is a regulated growth that is happening. Now, if you disturb the layer and take out some cells, then you will see that the cells still grow one over the top of the other if they are cancer cells. If they are normal cells, the moment they come in touch with the other cells and form a complete, if they fulfill, I mean if they fill this petri dish, they do not grow more than that. That is known as contact inhibition. The cancer cells do not have that contact inhibition, we call it loss of contact inhibition, and so they can grow one over the top of another, which is not the case with the normal cells. The other features of, we have just given a schematic diagram to understand some of the phenotypic differences between normal cell and a cancer cell. Like you have cytoplasm, nucleus, nucleolus, chromatin, all those things, but you can see in the nucleus it is full of this nuclear material. So many fragmented nuclei are there. So, that is typical, and also the cells are a little bit elongated than the normal cells. So, you can define that as increased nuclear cytoplasmic ratio and more DNA content and more mitotic figures. See, you see so much in the nucleus, so many dots are visible in the cancer cells. And variable chromosome number, that is typically shown in this electron micrograph that you can see in the bottom. See here, this is a normal cell, it looks like a flat one, whereas the cancer cells under electron microscopy, they look elongated and sharp tail-like structures are seen. So, I mentioned this loss of contact inhibition and variable chromosome number. As I told you, cancer cells have lot of mutations, they have genomic instability, they are characterized by genomic abnormalities and instabilities. The loss of contact inhibition, you call that for a different purpose, since the normal the normal cells, they depend on some surface to grow. For example, if you put them in a petri dish or in a test tube or whatever, they will grow, but they need that surface to grow. Whereas, cancer cells do not need that support. This is known as anchorage dependence. Contact inhibition is slightly different. Contact inhibition means when the cells come in contact with each other, they stop growing. That is known as contact inhibition. So, the cancer cells do not exhibit that. But in addition, the normal cells need some substrate or anchorage, that is also called substrate, some support to grow, whereas cancer cells do not need that. So, we call that as loss of anchorage dependence. And the cancer cells are unique in that they can secrete certain growth factors. This is very important to understand. See, the first cancer cell line was from an American woman, you know, it is called HeLa, you know that, right. So, that represents her name. So, that was discovered somewhere in, she had, she was an African American woman, she had cervical cancer, from whom the first cell line was isolated. It is called HeLa, that is the first human cell line to be grown in the lab, and that is grown even today. So, the cancer cells have this kind of an immortal property because they are anchorage independent. And the reason for this kind of a growth as against the normal cells is that they secrete their own growth factors, so they can continue to stimulate growth. But this is not the only reason. There are many other characteristic features which we will formally discuss a little bit later, and they also express growth factor receptors. So, these are some of the important differences. In the case of malignant tumor, some of the important features during the long journey of a normal cell to be transformed into a cancer cell, there are several steps involved. So, let us say this is a kind of normal cluster of cells, a slight abnormality happens, let us say a mutation happens, and then the structure slightly changes. There are some structural changes, you can see that. This initial condition is what is called hyperplasia. Just now I described the two terms. So, this is referring to the structural changes. When these changes are restricted to the site of origin and do not spread, then it is called in situ cancer. Then what happens? They slowly develop, multiply, and the first thing they have to do is to cross over the blood vessel. It can be lymphatic system or it can be simple blood vessels. So, they have to cut open that, then they will enter, then the job becomes easier, they can travel all over. Now they got a vehicle. So they will travel to distant sites and then settle down there. So this is a typical feature of invasive cancer cells. They become metastatic by this mechanism. So this is a simplified schematic to explain this, just for an understanding. The other interesting feature of cancer cells is most cancers occur in highly proliferating cells. You know, when you are walking and you are struck with a stone, you may slightly tumble, you may even fall down, but you may not have a big damage. But if you are running and you are struck by the same stone, then you know the consequences. It will be much more severe. So, it will be similar to that analogy. These analogies are given just to make you understand. So, we should not stretch it too much. So, proliferating cells have the tendency to become abnormal and then they may turn into cancer cells. The two important examples are epithelial cells and stem cells. Later on I will explain the stem cell theory also. Stem cells, you know what are stem cells. So, they can practically change into any other shape, right. So, they can, what are the other properties? They can proliferate, they can take the shape of other cells. So, from a fibrotic cell you may get a liver cell or whatever type of specialized cells. So, they can practically take different avatars, we call it pluripotent cells, right. So, people also think that they are the initiator cells for cancer. There is a theory which I will explain shortly. Or alternatively, this is one thing, highly proliferating cells. Other avenue is chemicals, viruses and environment. Just now we talked about the various factors that can induce cancer. So, they can also ultimately turn the normal cells into cancer cells. Now, I will ask one interesting question for which the answer is there in the next slide. You all heard of cervical cancer, breast cancer, stomach cancer, pancreatic cancer, many cancers you must have heard of. Have you heard of heart cancer? Heart can get cancer or not? Even recently there was a newspaper report here in Chennai that somebody got a huge tumor in the heart and it was removed. So, heart can get cancer, but it is very rare. It is not detected that much, it is a very rare type of cancer. Now, you can, but why it is rare? The question is, I just defined that highly proliferating cells get cancer. So, heart cells do not divide that much. They have, in technical terms, they have low mitotic index. It does not mean they cannot get cancer, they can, provided there are some issues with the signaling mechanisms. Some abnormalities if they happen, then they can also get cancer. Now, you have to ask me another question. Sir, brain cells also do not divide that much, but we have brain cancer, correct? Why is that? Can anyone think of it? While the actual neuronal cells are also like myocardial cells, they also do not divide that much, but there are, what do you call that? Glial cells, astrocytes, Schwann cells and so on. So, those cells are dividing. So, they can get, that is why the most common so called brain cancers are schwannoma, glioma and so on, astrocytoma and so on. So, they can get, even neuronal cells like heart cells in a rare situation can get cancer. Practically all the tissues can get cancer, but neuronal tissues and heart cells, it is somewhat rare. Rare does not mean it does not occur, it occurs, but rarely. That is what you have to, that distinction we have to understand. Here you see heart cancer listed as a rare disease in the US by the so called National Institute of Health, because it affects less than 200,000 people. In the, we can say 2 lakh, but in the US they will say 200,000 in the entire US population. So, cardiac sarcoma, rhabdomyosarcoma, angiosarcoma are some of the examples. It cardiac tissue can be affected. I asked this question already. We know that nerve cells cannot reproduce, then how do people get brain tumors? The thing is, other cell types like glia, astrocytes and Schwann cells, they can get cancer. So, that is the idea. Now, cancer cells, I already told you some properties, like they have loss of contact inhibition, they have loss of anchorage dependence, they are anchorage independent and so on. There are other hallmarks, at least 6 essential alterations acquired by cancer cells have been listed by experts. Weinberg and Hanahan, they have written a review long back, and again they have revised after 10 years, they have given other hallmarks also, but these 6 essential hallmarks are known to be acquired by the normal cells and they become cancer cells. By this you can also understand that the cancer cells are not alien cells, they have come from our own cells. This is one of the reasons why we are not able to treat them completely when they become so severe. Because one thing is they are nothing but some cells that have originated from the normal cells. So, multiple genes are affected, many genes are affected, and so which gene you will treat? If you start treating them, then you are also affecting the normal genes. So, this is why most all of the chemotherapeutic agents have side effects. Although we are giving them to treat or to kill cancer cells, they do affect normal cells, but with less proportion. That is why we are using, we are basically compromising. Right? You can ask that, sir, we know that these chemicals are poisonous, then why are we using them? We are using them as a matter of compromise. They do affect normal cells, that is why you get the side effects, loss of hair, loss of immunity, vomiting, nausea, so many side effects you see that. So, that is an important factor. So, when this transformation takes place from where a normal cell gets converted to a cancer cell, there are 6 essential alterations, physiological alterations. Number 1, self-sufficiency in growth signals. There is another fundamental thing that I want you to understand. In biology, you have certain signals. The cells do not function without receiving a signal. Any living organism, it needs a signal to do any function. So, that is an important thing for you to understand. Signals are very much necessary. There is another important concept that you may have to keep in mind, that is something similar to Newton's third law. For every action, there is an equal and opposite reaction, we say. So, for every signal you have some opposite signal. In other words, if there are, but they need not be equal and opposite. They are opposite, but need not be equal. They can be less than equal, somewhat equal, or sometimes equal, sometimes more than equal. So, there are different conditions that might turn up. So, the net result could be decided by the war between these 2 signals. For example, I will just tell you, I can go on that. For example, if there are activators, there are inhibitors. If there are inducers, there are repressors. If there are oncogenes, there are tumor suppressor genes, like that. So, for if there are pro-apoptotic genes, there are anti-apoptotic genes. So, like that you will find as we go along, and you must have studied already in cell biology and molecular biology also, enhancers, repressors, all those conflicting factors. So, it is actually a kind of tussle between all these things. That is why I said when I defined cancer in the beginning, it is a tussle between proliferation and cell death. I said it is not that it is only these two are the factors, but this is how it is. Opposing factors will play a role, and these opposing factors within a cell constitute what is called a microenvironment. Later on we are going to discuss that also. So, here the first point is self-sufficiency in growth signals. Just a few minutes back I described that the cancer cells have their own growth factors. So, they will have their own growth factor receptors. So, they will be able to stimulate themselves. So, self-sufficiency in growth signals. Another interesting feature is the second one, insensitivity to growth inhibitory signals. There are many growth inhibitory signals. For example, transforming growth factor beta signaling is an inhibitory signal actually. So, it is an inhibitory signaling pathway. Normal cells will obey to that, but cancer cells will not obey to that. So, insensitivity, they are not sensitive to growth inhibition. So, that is another characteristic feature, and related to that is the growth inhibition is one thing, cell killing is another thing. For example, in bacteria also we say that bacteriostatic or cytotoxic, cytostatic and cytotoxic we say. Static would mean arrest of the growth, but killing, toxic when we say, we are referring to the killing. So, similarly here also, when the cells are getting killed, there is a mechanism called apoptosis, which is called programmed cell death, about which also we will study in detail. So, the normal cells, supposing the normal cells get affected by DNA, by radiation, let us say. So, there will be a DNA damage. So, what they will do? They will try to repair that damage. Suppose the damage is too heavy, you cannot repair it, it has crossed a threshold. Many of the cells, they undergo apoptosis. In other words, they commit suicide, you can say, something like that. What is the difference between suicide and murder? Suicide, the person who wants to die decides himself or herself, whereas in murder somebody else decides that you have to die, right. So, similarly here also, the cells themselves, once they get some signals and they think that this is crossing the threshold, then the best way seems to be to commit suicide. That is what is programmed cell death. This is a very interesting topic. We will discuss that in detail later also, but understand that it is very important in multicellular organisms. Why? Because in a single cellular organism, that is the end of the story. In multicellular organism, it is the beginning of another story, because in multicellular organisms, this is a protective mechanism, because the cells prevent other daughter cells from getting this DNA damage. So, apoptosis is very important. Apoptosis is important in many other aspects also, which we will discuss later, but this is one example. So, normal cells, if they get this kind of a signal, they will obey that and undergo apoptosis, but cancer cells will not. They want to live. I am telling for the sake of understanding. What happens is they have defective apoptosis signaling, and so they will not undergo apoptosis, and thereby they will live longer. That is what they want also, right. They want to propagate, they want to proliferate. So, that is it. So, evasion of apoptosis. And there are defects in DNA repair. Yes, DNA repair is a unique story. If there is a damage to the DNA, the cells will try to repair. There are several mechanisms which again we will discuss in detail later on, whereas in cancer cells there will be, actually what happens is the anti-cancer agents, they do DNA damage and thereby they kill the cells. So, the DNA repair mechanism is 1 over. So, DNA repair mechanisms will fail ultimately, because they can do only to some extent, there is a threshold. So, that threshold is crossed over when you are giving some chemotherapy or some radiation or whatever. So, they do extensive DNA damage. So, the cells are not able to handle that. So, by producing heavy damage, they kill the cells, just like apoptosis I told you. Then there is limitless replicative potential, we will talk about that later again, and the cancer cells require blood supply, that is called sustained angiogenesis. And then invasion and metastatic properties are the notorious properties. So, you can count 1, 2, 3, 4, 5, some 6 are there. Invasion, if you leave, then there are 6 hallmarks. Invasion is again, it becomes metastatic. So, there are 6 hallmarks, that is what people say. But then after this review was written, the other scientists came to some discussion, and then they said, why you are leaving the immune system? Immune system normally protects us, but under certain conditions it can also enhance carcinogenesis. I will talk about it later. When chronic inflammation, under some conditions they can be turning against us even. But avoiding immune destruction is an important mechanism. So, that is also another, because immune cells protect us. So, the cancer cells have to avoid that immune protection. So, that is one mechanism, they do that. Then deregulating cellular energetics, that is also another mechanism. Genome instability and mutation are characteristic features. We are talking about the different hallmarks of cancer cells. And then tumor promoting inflammation. As I told you, chronic inflammation will be tumor promoting. So, the acute inflammation will not be, that will be a kind of protective, but chronic inflammation will be promoting the tumor formation. So, these are thought to be some of the emerging hallmarks and enabling characteristics of cancer cells. So, this is just basically these slides are introductory in nature, and I am just giving you some overall view before going into other details. Just now I mentioned that tumor microenvironment. See, when we study cancer cells, people often are very critical about studying the cancer cells in vitro. For example, in my lab I grow the cancer cells in vitro, tissue culture or cell culture, whatever you want to call. So, we grow the cancer cells and we can use them for studying several mechanisms. For example, if you have an anti-cancer, if you think something is an anti-cancer agent, you cannot just give it to some people and then test it out, right. So, you test it out on cancer cells and extrapolate that findings to humans, that is what we do. So, people are very critical about it, because what is true for these isolated cells, they are not normal, you see, you have isolated them from the body, human body. So, they are not anymore with the body. In the body, they are surrounded by so many other cells, whereas in the lab you are only isolating them separately and then growing. How come any outcome that you are getting will be applicable to human? That is a very relevant question. It is true also, it is not always applicable, but that knowledge over the years, history tells us that knowledge gained through these systems and lower organisms have helped us to formulate several theories, and even for the discovery of several drugs, it has turned out. So, history taught us that doing experiments on these ultimately helps. So, it is a collective effort if you want to get into drug development. Anyway, here the point is, the cancer cells alone are not the culprits. For a normal cell to get transformed into a cancer cell, it has to undergo several changes. So, how this happens in the body, in the organism? It is surrounded by several cells. There is a saying, you tell about your friend, I will tell about you. So, there are influences from others. Similarly, here also the other cells secrete several factors which influence whether the normal cells have to be transformed, or they may have suppressing factors also which may prevent them from turning into cancer cells. So, it is all a collective effort of all these. Ultimately, it turns out that they turn into cancer cells. So, what are those cells? This is what is called a microenvironment. When it turns into a tumor, you call it as a tumor microenvironment, which facilitates the formation of tumors. So, what are the cells? The cancer cell is there, lymphocytes. There are some cancer cells, some normal cells at any given point of time. Even during surgery, that is one of the difficulties. If you want to remove the tumor alone, it is not that tumor is there as a tumor. In between the tumor, inside the tumor, a large mass, there will be some normal cells also. Ideally, you do not want to remove the normal cells. So, by any method, it is difficult to separate them out. Surgery also, including surgery. What they do is they remove everything, so that you get rid of some normal cells also, you sacrifice. They can do that. In fact, they will do some margins and some extra margins and then can remove. Of course, there are a lot of improvements over the years, but this is what is the principle. So, it is very difficult. They are always surrounded by other cells. So, cancer cells are there, lymphocytes, and then endothelial cells, and what are known as pericytes, and some fibroblasts, and what are known as myofibroblasts also, number of cells, neutrophils, macrophages, and what is shown or not shown there are the stem cells also. They are also very important. Some of the progenitor cells, stem cells, these will ultimately influence the course that the normal cell has to take, whether it has to develop into a cancer cell or need not develop into a cancer cell, stuck up there, be dormant. So, this is all decided by this microenvironment. It is a very important thing. So, the question comes, do we know what are the changes in this long journey of a normal cell into a cancer cell? The answer is, each cancer patient is different, and so what is happening with everybody we cannot know. We can only generalize certain things. It is not known for many of the cancers. What are the sequential events? What are the genes that are mutated? Which genes are mutated? What are the genes which are not mutated? What is the next step? And so on. So, it was thanks to efforts from Bert Vogelstein's lab in the US, they have found out certain sequence of events. You remember I told you in US, one of the most common cancers is colorectal cancer. So, a lot of research is going on colorectal cancer. So, one such lab by Bert Vogelstein, they found out certain changes that commonly happens in colon cancer patients. Suppose a normal colon cell is there, chromosome 5 gene is first affected, then there is increased cell growth, and then DNA loses some methyl groups. Later on I will tell you about the epigenetic changes. Methylation and methylation plays an important role. So, one of the epigenetic changes takes place that develops what is called low level polyp. Polyps are protrusions in the colon, small protrusions, and then you get RAS. RAS is an oncogene. So, RAS gene mutation is one of the first steps, and then there is also a loss of certain genes in chromosome 18, it is called DCC, deleted in colon cancer. The gene name itself is like that, DCC, deleted in colon cancer. Then that leads to further deterioration of the situation. it develops high level polyps. And then in chromosome 17 there is a loss of p53 is a very well known tumor suppressor gene. We will talk about that. There is a loss of p53 gene and then you get a full fledged cancer cell. Then you need additional changes, chromosome losses, for these cancer cells to become metastatic. So, these are the somewhat well defined sequence of changes for any known cancer, colon cancer. For other cancers, do we have this kind of a step? Almost no. So, we only know a little bit about the sequence of changes. This also you cannot take it as verbatim. These changes are largely observed. So, we take it like this. So, the invasion and metastasis is very important. It is sometimes called invasion metastasis cascade. There are several steps involved in that. The first thing is the formation of a primary tumor in a tissue, and then first it has to invade in the neighboring tissues or in the blood. That is called localized invasion. That is one of the first steps before the cells become metastatic. The second thing is interaction. Once this kind of entering into the blood vessel is called intravasation. Vasation, vessel, it all refers to blood vessel we say. So, it is called vasation, intravasation, intra means inside. So, getting inside the blood vessel is called intravasation, it is very simple. Getting outside is called extravasation. So, once it enters, it pierces the cells. Basically what happens is in the cell extracellular matrix, those enzymes are proteases and all that, matrix metalloproteases will be involved, and they will digest the proteins on the outside, and thereby they can make holes into those cells. So, then naturally the blood component, blood by the way is a tissue, it consists of several cells, red blood cells, platelets, lymphocytes, all kind of blood components, it has to interact with them. Then next is of course they have to travel throughout the circulation. Then they get arrested in some organs. This is a very interesting phenomenon. They get arrested, they get stopped during the blood supply, blood is taken to different tissues and then they stop at specific tissues. Interestingly, it is the lungs, bone, brain are very well known secondary metastatic sites. Then what happens? They have to come out of the blood vessel, that is called extravasation. Then only they can land up in these tissues. So, that is known as extravasation. Then once they come out and join a place, then they have to colonize that place, that is called micrometastasis. Then it slowly develops and forms a bigger colony, it is called macrometastasis. So, these are the various steps involved in the invasion metastasis cascade as I told you. So, I have listed out that 6 steps to metastasis, just like 6 hallmarks, I just put it the same 6 number just to remember. Breach of basement membrane, intravasation, transport to distant tissues, arrest at what is called the target organs or secondary metastatic sites, and extravasation, formation of micrometastasis and colonization at distant sites. There is a slight confusion here, perhaps you may think extravasation and arrest at target organs. It is not like that. When they are going to a specific area in the blood supply, they stop in that organs and now they continue to penetrate to that organ. That is why it is given like that, and extravasation. They have to come out of the blood vessel I will tell you some of the mechanisms also. So, the basic routes are the lymphatic spread. Although tumors have no lymphatic supply by themselves, surrounding areas have lymphatic vessels. So, most common way by which they spread is through the lymphatic route. Breast cancer and axillary lymph nodes. If you go to a doctor, the first thing they will look at is the lymph nodes, the axillary lymph nodes or many other lymph nodes here and so on. Because if the cancer has spread to lymph nodes, that is an indication that it is going to be metastatic. So, they look at lymph nodes and look for cancer cells there. In fact, they may take a biopsy and look for cancer cells in those lymph nodes. So, lymphatic spread is the most important route, and of course, through blood supply. Liver, lungs, bone marrow are the most commonly involved secondary metastatic sites, that you can call that as hematogenous spread, hemato referring to blood, extensive because of extensive blood supply. Veins are more easily penetrated because vein, that wall is thin compared to artery. Arteries are thick walls. So, veins have thinner walls, that is why they can penetrate through veins. Cancer cells are larger than the red blood cells and white blood cells and not easily deformable. They get trapped in capillary beds, especially in the lungs. Because they are somewhat bigger cells, the capillaries are small vessels, so they can get stuck up compared to other normal cells. These are slightly bigger, and so they get stuck up in small capillaries, especially in small capillaries and in the lungs. Now, the question comes, what cancers are spreading to what tissues? I have given a table so that you can remember. We will take up prostate cancer, which is happening in the men. Preferred secondary site, of course, the first site is prostate gland. Prostate is one organ that grows as you become older. Normally, organs will not grow as you become older, but this is one exception. Especially the elderly men will have a larger prostate. Even if they do not have cancer, their prostate will be slightly enlarged. But that is one cancer affecting men, and the secondary site is bone marrow. Other secondary sites are liver, lungs and brain, but the major secondary site is bone marrow. Breast cancer, again it can spread to bone marrow as well as brain, lungs, liver and so on. The primary site is breast. Colon cancer, it spreads to liver or lungs, bone marrow. I have given the preferred site in the second column and the other sites given in the second column, so that it is easier for you to remember. Pancreas to liver and then to lungs. So, these are the important cancers, and this phenomenon of spreading to specific tissues is called metastatic tropism.